Hello everyone. So in this video, we're going to talk about the histology of eye and ear. So for eye is a complex sensory organ that provides the sense of sight, and our eye measures approximately about 25 millimeter in diameter. So it's suspended in the bony orbital socket by 6 extrinsic muscles that control its movements, and also a thick layer of adipose tissue partially surrounds and cushions the eye as it moves within our orbit. And the extraocular muscles are coordinated so that our eyes can move symmetrically around their own central axis. So as for the layers, our eye consists of 3 concentric layers, or coats we call. So the first layer is corneoscleral coat. So this is the outer or fibrous layer that includes sclera, the white portion, and the cornea, also the transparent portion. And the next layer is vascular coat, also called uvea. It's the middle layer and includes the choroid and the stroma of the ciliary body and iris. And the last part is the retina. It's the inner layer and it includes an outer pigment epithelium and the inner neutral retina and the epithelium of the ciliary body and iris. So the neutral retina is continuous with the central nervous system through our optic nerve. Now let's move on to the specific information for corneoscleral coat. So the corneoscleral coat consists of the transparent cornea and white opaque sclera. The cornea covers the anterior 1/6 of the eye, and in this window-like region, the surface of the eye has a prominence or convexity. The cornea is continuous with the sclera, and the sclera is composed of dense fibrous connective tissue, and it provides attachment for the extrinsic muscle of our eye. So for the vascular coat, or uvea, the uvea consists principally of the choroid and the vascular layer that provides nutrients to the retina. So blood vessels and melanin pigment give the choroid an intense dark brown color, so the pigment absorbs scattered and reflected light to minimize the glare within our eyes. The choroid contains numerous venous plexuses and layers of capillaries and is firmly attached to the retina. And the anterior rim of the UV layer continues forward, and it forms stroma of the ciliary body and iris. And the ciliary body is like a ring thickening that extends inward just posterior to the level of cornea-scleral junction. And within the ciliary body, there's a ciliary muscle. The iris is the contractile diaphragm that extends over a smooth muscle that is responsible for less accommodation and anterior surface of the lens. It also contains smooth muscle and melanin-containing pigment cells scattered in the connective tissue. So as for retina, the retina is a thin, delicate layer and it consists of 2 components. And the neural retina is the inner layer that contains light-sensitive receptors and complex neuronal networks. And the retinal pigment epithelium, also called RPE, is the outer layer that is composed of simple colloidal melanin-containing cells. And externally, the retina rests on the choroid, but internally it's associated with the vitreous body. The neural retina consists largely of photoreceptor cells, which are called retinal rods and cones, and interneurons. So as for the chambers, the layers of the eye and the lens serve as boundaries for the 3 chambers within our eyes, and there's anterior, posterior, and vitreous. So the anterior chamber is the space between the cornea and the iris, and we can see it at the top of this picture. And the posterior chamber is the space between the posterior surface of the iris and the anterior surface of the lens. And in this picture, we can see it on the left side. The vitreous chamber, which is this part in the middle, it's a space between the posterior surface of the lens and the neural retina. The cornea, the anterior and the posterior chambers and their contents constitute the anterior segment of the eye. The vitreous chamber visual retina, RPE, and posterior sclera and uvea constitute the posterior segment. Moving on to the development. So our eyes forms from neuroectoderm, surface ectoderm, and mesoderm. So by the day 22, shallow grooves called optic sulci appear and it becomes optic vesicles. Then the optic cup with two layers are inner layer and outer layer, and the inner layer becomes the neural retina, whereas the outer layer becomes the retinal pigment epithelium. The lens forms from the surface ectoderm as the lens vesicle, which later separates and sits inside the optic cup. And that surface ectoderm also forms the corneal epithelium, while the mesoderm forms the corneal stroma and endothelium. The choroid fissure forms below the optic cup, and it allows the hyaloid artery, and it later becomes the central retina artery and vein. And then, by the week 7, our pupil forms as the fissure closes. The retina starts forming layers by week 5, and by month 7, key cells like rods, cones, and ganglion cells are present. The fovea forms in the 8th month and matures after birth. And by month 3, the ciliary body and iris develops, and they both come from the optic cup, and the iris, both layers are pigmented, and in the ciliary body, only the outer layer is pigmented. And the iris vessels form from the neuroectoderm by month 6, and at birth, the iris is often light blue, especially in lighter skin tones, because of its low pigment. So let's talk about the microscopic structures of our eye. So let's start with the corneoscleral coat. So the cornea consists of 5 layers, which are 3 cellular layers and 2 non-cellular layers. The transparent cornea is about only 0.5 millimeter thick at its center and about 1 millimeter thick peripherally. And it consists of 3 cellular layers that are distinct in both appearance and origin. And those layers are separated by 2 important membranes. And the 5 layers of the cornea and transverse sections are corneal epithelium, boneless membrane, corneal stroma, the cement membrane, and corneal endothelium. So, for corneal epithelium, the outermost layer is non-keratinized stratified squamous epithelium, and it's about 50 micrometers thick with approximately 5 cell layers, and it regenerates very rapidly. It's usually between 7 days from corneal limbal stem cells, which are located at the corneoscleral limbus. And for the Bowman's membrane, this is about 8 to 10 micrometers thick, and the cellular layer lies beneath the epithelium and provides structural support and a barrier to infection. And it consists of randomly oriented collagen fibrils and does not regenerate if damaged, and scarring of this layer can impair vision, and it's associated with the recurrent corneal erosion, and numerous free nerve endings provide high sensitivity. And for the third layer, it's corneal stroma, and it makes about 90% of the corneal thickness, and the stroma consists of 60 lamellae of orthogonally collagen fibrils interspersed with keratinocytes, and transparency is maintained by uniform peripheral spacing, and they're regulated by proteoglycans like lumican and decorin. And swelling or injury disturbs the structure, and they lead to opacity and impaired vision. And as for the cement membrane, this posterior basal membrane supports the corneal endothelium, and it's about 10 micrometers thick and increases with age. And it has felt-like fibrous structure and stains strongly with PAS and can regenerate after injury. Extensions of this membrane form the pectinate ligament, helping maintain corneal curvature. This is a single thin layer of flat cells on the inner surface of the cornea, facing the front chamber of the eye, and it controls the amount of water in the cornea to keep it clear by using special pumps that provide sodium and potassium. and these cells don't divide easily, so if they are badly damaged, a cornea transplant might be needed. So for the next structure, it's the vascular coat, or uvea. So it's the middle layer of the eye and has three main parts, which are iris, ciliary body, and choroid. So iris is the color part of the eye that controls how much light gets in, and it has two muscles, which are sphincter pupillae, and it makes the pupil smaller in bright light, and dilator pupillae, and it makes the pupil bigger on dim light. And ciliary body. So behind the iris, it's behind the iris, and it makes the aqueous humor, and it helps focus the lens and holds it in place with zonular fibers. It has three muscle groups, which are longitudinal, radial, and circular. The longitudinal stretches the choroid, and the radial one flattens the lens for far vision. The circular one thickens the lens for near vision. So the choroid, it's the back part of the uvea, and it's packed with blood vessels that feed the retina. It has two main layers, which is choricapillary, and those are small vessels that feeds the retina, and Bruch's membrane. It's a thin barrier between choroid and retina. So the last part is aqueous humor. Aqueous humor is a clear liquid and the fluid made by the ciliary body, and it nourishes the cornea and lens and keeps the eye pressure stable, and it drains through the trabecular meshwork and canal of Schlemm. So let's move on to retina. So the retina is the inner layer of the eye, and it has two main parts. And the neuroretina, which is the inner part, contains photoreceptor cells, which are rods and cones that detect the light, and retinal pigment epithelium, which is the outer part, and they support the photoreceptors and is attached to the choroid. And these two layers can separate, causing retinal detachment, and the retina has a non-photosensitive part, which is in the front of the eye, over iris and ciliary body. And the photosensitive part is in the back of the eye, and this is where the vision occurs. So there are few important regions, which are optic disc, and this is where the optic nerve exits the eye. No photoreceptors means a blind spot. And the fovea centralis. It's a small central pit with the sharpest vision. And there's macula lutea. It's a yellow area around the fovea, and key for detailed central vision. And also it can be affected by macular degeneration as we age.
Now, let me walk you through the 10 histological layers of the retina from the outermost to the innermost. The first layer is the retinal pigment epithelium, or RPE. It consists of a single layer of cuboidal cells containing melanin to prevent the light scattering. It also phagocytoses the outer segments of photoreceptors, regenerates visual pigments, and forms the blood-retinal barrier. The second layer is the photoreceptor layer, which contains the outer segments of rod and cones. These cells detect light and convert it into electrical signals. This is where visual transduction begins. The third layer is the outer limiting membrane. It is a boundary formed by junctions between Muller cells and photoreceptors. Though not a true membrane, it provides a structural separation within the retina. The fourth layer is the outer nuclear layer, which contains the nuclei of rod and cone cells. The thickness of this layer varies depending on the density of photoreceptors. The fifth layer is the outer plexiform layer, where photoreceptors synapse with bipolar and horizontal cells. This is the site of the first synaptic transmission in the retina. The sixth layer is the inner nuclear layer. It contains the cell bodies of bipolar, horizontal, and amacrine cells, along with Muller glial cells. It is where complex signal processing begins. The seventh layer is the inner plexiform layer. In this layer, bipolar cells forms synapses with ganglion cells and amacrine cells. This is a key site for visual signal integration. The eighth layer is the ganglion cell layer. It contains the cell bodies of ganglion cells that form the optic nerve and transmit final visual signals to the brain. The ninth layer is the nerve fiber layer. It composed of axons from ganglion cells that run horizontally and converge to form the optic nerve. The 10th layer and final layer is the inner limiting membrane. It is formed by the basement membranes of Muller cells and serves as the boundary between the retina and the vitreous body. Next, let's compare the two types of photoreceptor cells in the retina, rods and cones. The photoreceptors of the retina consist of rods and cones. Rods are highly sensitive to light and allow vision in dark environments, using a single pigment called rhodopsin. Cones, in contrast, function in bright light and are responsible for color vision. They contain three types of pigments: red, green, and blue, and are concentrated in the fovea, supporting high-resolution central vision. Now, let's explore how light is converted into electrical signals by photoreceptors. When light reaches the retina, 11-cis-retinol inside photoreceptors is converted into all-trans-retinol. This activates opsin, which then activates the G protein transduction. Transduction stimulates phosphodiesterase, reducing cGMP levels and closing sodium channels. The cell becomes hyperpolarized, glutamate release decreases, and the signal is passed on to bipolar and ganglion cells. Let's look at two key anatomical landmarks of the retina, the fovea and the optic disc. The fovea is located at the center of the retina and contains only cone cells, providing the sharpest vision. In contrast, the optic disc lacks photoreceptors and serves as the exit point for the optic nerve, creating a natural blind spot. Finally, let's examine the structure of the lens and its role in focusing light. The lens reflects light to focus it onto the retina. Its interior surface is covered by simple cuboidal epithelium, while the anterior consists of elongated lens fiber cells that lack nuclei and organelles. With aging, the lens loses electricity, leading to presbyopia, and protein changes can result in cataracts. Now let's move on to the ear.
The inner ear, also known as the labyrinth, is a complex sensory organ located in the petrous part of the temporal bone. It plays a critical role in both hearing and balance. Structurally, it has two main parts, the bony and the membranous labyrinth. The bony labyrinth is made out of interconnected cavities, the cochlea, the vestibule, and the semicircular canals. These are filled with perilymph, a fluid similar in composition to extracellular fluid, high in sodium and low in potassium. Suspended within the bony labyrinth is the membranous labyrinth, which contains structures lined by epithelium and filled with endolymph, a potassium-rich fluid essential for sensory transduction. They work together to support both auditory and vestibular functions. The membranous labyrinth is also divided into two parts, the cochlear labyrinth and the vestibular labyrinth. The vestibular duct contains the semicircular duct, the utricle, and the saccule. The utricle and saccule are connected by the utricular-saccular duct. The cochlear labyrinth contains the cochlear duct, which is inside the cochlea and continues from the saccule. There are 6 sensory regions in the membranous labyrinth that contain the special sensory and supporting cells. The 6 regions are in 3 cristia ampullaries of semicircular duct, 2 macules of utricle and saccule, and lastly the spiral organ of Corti located in the cochlear duct. The hair cells of vestibular and the cochlear labyrinth function as mechanoelectrical transducers, meaning they convert mechanical energy into electrical energy that can be transported by the nervous system. On the apical surface, there is a hair bundle which is made out of rows of stereocilia. The rows are called sensory hair. In the vestibular labyrinth, each hair cell has a true single cilium called the kinocilia. The stereocilia surrounds the kinocilia. The kinocilia is usually next to the longest rows of stereocilia. The stereocilia is made out of actin filaments cross-linked by fimbrian and espin-based connectors. At the top of the stereocilia, there is a mechanoelectrical transducer channel proteins. This channel is opened when the stereocilia moves towards the kinocilia. This channel opens and it causes polarization. Type 2 hair cells are cylinder and have both afferent and efferent nerve endings, while the type 1 hair cells are flask-shaped with usually afferent nerve endings. Each ampulla of semicircular duct contains a crista ampullaris, which is receptor for angular movement of the head. The crista ampullaris is a thickened transverse epithelial ridge that is oriented perpendicularly to the long axis of the semicircular canal. There are hair cells and supporting cells. Cupula, a gelatinous mass, is attached to the hair cells of each crista. Deflection of the stereocilia in the narrow space between the hair cell and the cupula generate nerve and impulses in the associated nerve endings. In picture 2, we can see hair cells in the upper layer and supporting cells in the basal layer. This epithelial layer is covered by the cupula. The epithelial layer is also innervated by nerves that come from the temporal bone. The macula of the saccule and utricle are innervated sensory thickenings of the epithelium that face the endolymph of saccule and utricle. Each macula consists of type 1 and type 2 hair cells, supporting cells, and nerve endings associated with the hair cells. Hair cells are polarized with respect to the striola, an imaginary plane that curves to the center of each macula. The gelatinous polysaccharide material that overlies the macula is called the autolytic membrane. The autolytic membrane moves on the macula in a manner analogous to that by which a cupula moves on the crista. Movement of the autolytic membrane causes the stereocilia to move towards the kinocilium, activating the MET channels. This depolarizes hair cells and generates action potential. Displacement of stereocilia in the opposite direction away from the kinocilium causes hyperpolarization of the hair cell and inhibits generation of action potential. In picture 2, we can see autolytic membrane covering the hair and the supporting cells. Under the supporting cells, there are connective tissue and nerve fibers. The cochlear duct divides the cochlear channel into 3 parallel compartments called the scala. The 3 compartments are media, vestibuli, and tympani. The cochlear duct itself is the scala media. The scala vestibuli and scala tympani are the spaces above and below, respectively, the scala media. The scala media is an endolymph containing space that is continuous with the lumen of the scala and contains the spiral organ of Corti, which rests on its lower wall. The scala vestibuli and scala tympani are perilymph containing spaces that communicate with each other at the apex of cochlea through a small channel called the helictreme. The upper wall of the scala media, which separates it from the scala vestibula, is the vestibular membrane. The lateral or outer wall of the scala media is bordered by a unique epithelium, the stria vascularis. It is responsible for the production and maintenance of endolymph. The stria vascularis encloses a complex capillary network. The lower wall or floor of the scala media is formed by a relatively flaccid basilar membrane that increases in width and decreases in stiffness as it coils from base to apex of cochlea. In picture 2 of the histological slide, we can see a spiral ganglion that originates from the cochlear nerve go into the scalar media. The spiral lamina and the vestibular membrane can be seen covering the roof of scalar media. The lateral is the stria vascula covering the scalar media. The spiral ligament and the basal membrane separates the scala vestibular and the scala tympani. The spiral organ of Corti is a complex epithelial layer on the floor of scalar media. There are inner and outer hair cells, inner and outer phalangeal cells, and lastly the outer and inner pillar cells. The inner hair cells form a single row of cells throughout the cochlear duct. The number of cells forming the width of the continuous row of outer hair cells is variable. 3 ranks of hair cells are found in the basal part of the coil. Phalangeal cells are supporting cells for both rows of hair cells. The phalangeal cells are associated with the inner hair cells surrounds the cell completely. The apical end of the phalangeal cells are tightly bound to one another and to the hair cells by elaborate tie junctions. These junctions form the reticular lamina that separates the endolymphatic compartments from the true intercellular spaces of the organ of Corti. The extracellular fluid in the intercellular space is called Corti lymph. Pillar cells have broad apical and basal surface that forms plates and narrow cytoplasm. The inner pillar cell rests on the tympanic lip of the spiral lamina. The outer pillar cells rest on the basal membrane. Between them they form a triangular tunnel, the inner spiral tunnel. In picture 2, the organ of Corti begins as a cochlear nerve and enters the basal lamina, and the formation of limbus spiralis starts to show. Inner border, inner phalangeal, inner hair, and inner pillar cells are all located near the limbus of spiralis. The sequence of cells are reversed on the outer border until it ends with the outer border cell, or the cell of Hensen. Hair cells are the receptors, while all the other cells are supporting cells.
Thank you.